Hello everyone, welcome to Nobel Forensics. Here I am explaining about the forensic hair evidence. In part 1 and part 2 I explained about the basics of hair and structure and growth cycle of hair. In part 3 of hair I am explaining about the examination and identification of hair and also differences between human and animal hair. Hair examination. There are two main types of forensic hair examination and they are hair identification and hair comparison. In the hair identification, identifying the particular hair like it is a human scalp hair, human pubic hair, human axillary hair, that is the hair identification. In hair comparison, comparing the hair like for example in the hair identification the particular hair was identified as human pubic hair. Then in the hair comparison, comparing that questioned pubic hair with the known standard pubic hair of suspect. That is in hair comparison, and this hair comparison is mainly based on color, structure and treatment of the hair. There is some elements for hair identification and there is 6 elements for hair identification. What are they? Questioned hair, standard or reference hair, literature references, equipment used, list of hair characteristics and the examiner. These are the 6 elements for hair identification. Very first one is questioned hair. The hair to be identified means which was found at the crime scene as an evidence. That is the questioned hair. And properly collected and packed, then sent to the laboratory, then they do analysis. That is questioned hair. And standard or reference hair, standard reference collection. Standard collections are prepared which contain large number of hair samples of different types and origin. Not only human hair but also animal hair. So we may not get animal hair at one place. So collecting from the zoos for different animal hair and also sometimes exchanging with other forensic scientists for standard or reference hair. Then literature references means the details about that particular hair. If cat hair, then the features of the cat hair, that is references. Literature references of hair also includes published photo micrographs like this. See, there is human hair, dog, deer, rabbit, cat, mouse. These are the photo micrographs of the hair. Then equipment used. There are different equipments are used for hair analysis and mostly important one is the comparison microscope. You all know that comparison microscope is used for comparing the two evidences side by side. Here for hair comparison. And you all know that comparison microscope means two compound microscopes joined by a common bridge. And then scanning electron microscope. The scanning electron microscope SEM, which is used to view hair scale patterns, cross-sectional shapes and medullary structures. And this is the scanning electron microscope and this is the cross-sectional shape of hair. And you all know that comparison microscope and scanning electron microscopes are so expensive. So we may not use always, but the same information can be revealed by using light microscope but with using some special techniques. See, this is the light microscope. Some special techniques are used to examine the hair sample under the light microscope for getting the same information like scanning electron microscope and comparison microscope. Then for example, I added one thing here, preparation of cast impressions, which means cuticular scale patterns. Cast impression of the hair scale patterns are produced by different methods and for this I added procedure taking 3 parts of the colorless nail polish and 1 part of the isoamyl acetate and mixing them properly and taking that mixture on a glass slide and spreading. Then after placing a hair sample on it and dry it for 10 minutes, then remove the hair sample. Then there you will get the cuticular scale pattern of the hair. Then observe it under the microscope. It is ready for the microscopic examination. You can view under light microscope that cuticular scale structure. Then that hair what we used for this cast impressions we can also use for further examination with simply washing in isoamyl acetate. Then list of hair characteristics. These are arise from the basic structure of hair, from the questioned hair, cuticle scales. What are the type of cuticle scales? I mean cuticle scale pattern. Then medulla, present or absent. If present, then what type? Fragmented, continuous, discontinuous. Then medullary index, whether it is more than 0.5 or less than 0.3. If it is less than 0.3, then it belongs to human. If it is more than 0.5, then it belongs to animal. Then there is any cosmetic treatment, any dyes applied, then whether it is cut hair or plucked hair. If it is plucked hair, then there is whether any chances to identify blood group and DNA analysis. These are the list of hair characteristics to be analyzed from the questioned hair. Then last one but most important one, the examiner. He synthesizes all information gained from various characteristics and develop a pattern. But for hair analysis, the examiner should have some features. He should be trained and experienced. Then there is no hard and fast rules to follow by examiner, and general characteristic patterns are important for hair examination. And in hair identification, an examiner should strive to obtain maximum information and give a well stated conclusion. And these 6 are the elements of hair examination. Then going to the human hair and animal hair differences. Here I added characteristic feature and humans and animals. Cuticle. In humans, cuticle scales are small, flat and are imbricate type and surrounded the shaft completely. But in animals, scales are large, polyhedral, wavy and do not surround the shaft completely. Then medulla. Narrow medulla may be absent sometimes. If present, it may be narrow, fragmented or discontinuous. But in animals, medulla is always present and also broad and continuous. Cortex. Cortex is thick in humans, 4 to 10 times broad as medulla. But in animals, it is thin, rarely more than twice the breadth of medulla. Medullary index. What is medullary index? Diameter of the medulla by diameter of the whole hair shaft is the medullary index. If it is less than 0.5, then it is humans. In animals, it is more than 0.5. Pigment. Pigment in humans more towards the periphery of cortex, but in animals, it is peripheral or central and it is uniform. These are the features of human hair and animal hair. Of course, differences of human hair and animal hair. In identification of animal hair, there are some animals and their features here. Some major identification features of animal hairs commonly occurring in forensic cases. Canidae. Canidae means dog hair. Hair usually has a complex cellular medulla with donut shaped cells in dog hair. Then coming to the cat hair. Cat hair is Felidae. Also has medulla with donut shaped cells, but the difference between dog hair and cat hair is medulla shows sudden taper at the end. Hair usually exhibits distal banding. That is the difference between dog hair and cat hair. Both dog hair and cat hair are having donut shaped medulla, but dog hair is having continuous donut shape. But cat hair, there is sudden taper at the end. Then deer hair means cervidae. It is having broad cellular medulla with a cobblestone effect. Deer hair having cobblestone effect. Then bovidae, cattle hair, cortex with large ovoid bodies, medulla is narrow and can be absent, fragmental or continuous. In the cattle hair, medulla may be absent or if present, fragmental or continuous. Then equidae, horse hair, hairs show transverse stria and scanty pigment irregularly distributed. In the rabbit hair, which is called leporidae, hair have a multiserial ladder type medulla and dog bone shaped cross section. It is having ladder type of medulla and also its cross section is dog bone shape. These are the different animal hairs. Along with microscopic and macroscopic animal hair identification, DNA and electrophoretic methods for species identification are being used in modern times. Not just with the characteristics of the hair, but also with the DNA and other electrophoretic methods, we can identify species, origin of species. Then human hair identification. If the hair was identified as a human hair, then what we do next? Then we identify some other features, whether it is scalp hair, pubic hair, axillary hair, means body area of origin. If it is scalp hair, usually long with constant diameter and having cut tips or flat tips. Then it shows sometimes cosmetic treatment, and also the scalp hair cross section is round to oval. Then usually scalp hair are the longest hair comparing with the other body hair. Then pubic hair. Pubic hair is usually kinky and wide variation in diameter. Tips are rounded or frayed because due to rubbing with the clothing. And also pubic hair is having broad medulla, usually Pubic hair cross section is kidney shaped. And beard or mustache hair, it is having broad diameter with a triangular cross section. Then eyebrow, eyelash or nozzle hair. They are usually short, stubby, tapering to an abrupt point. Usually these eyebrow, eyelash hairs are having mascara and nozzle hair having mucus adhering to the hair. Then usually they have taper ends, sharp ends. Then axillary hair resembles pubic hair, but the difference is they are less kinky and present a bleached appearance distally. And general body hair, they are having different combination of characteristics, so it is difficult to identify as a body hair. These are the different features of different hairs on the human body. Then method of removal means we can identify the hair whether it was cut hair, plucked hair or naturally fallen off hair. If a root sheath is present and the hair is in anagen growth phase, an examiner can conclude that the hair is indicative of forcible removal. Means when it is in anagen stage, then it is in active growth phase, so it cannot easily fall off. So if somebody plucked out only, then it will be come out of the follicle. So there is some forcible removal. Then a telogen root without a sheath indicates that the hair fell out naturally. Then if the root is not present, an even break with regular edges, which means it is a cut hair. Racial origin, identifying the race of the human. There is 3 races: Caucasian, Negroid and Mongolians. Then features, color. In usually Caucasians, it is color is variable and non-black color. And in Negroids, it is more black or brown color. But in Mongoloids, black or dark brown color. Pigment distribution. In Caucasians, evenly distributed. In Negroids, medium to coarse pigment granules with clumped pigment. In Mongolians, coarse pigment granules show high pigment density. Cuticle. It is the outer layer of the hair. Caucasians having thin to medium cuticle. In Negroids, medium to thick cuticle. And Mongolians, thick cuticle. Then cross section of the hair, Caucasians having oval shape cross section, Negroids flat cross sectional shapes, Mongolians having round cross sectional shape. Then diameter along the shaft. In Caucasians only there is slight variation among the diameter of the shaft. In Negroids apparent wide variations. Mongolians constant diameter along the length of the hair. These are some racial features of the hair. Caucasian, Negroid and Mongolians. Cosmetic treatment. There are various examinations that are done to detect cosmetic content of the hair. Here added few microscopical examination. The colors in the cuticle provide indication of dyeing. Hair growth rate combined with measurement of untreated portion can help in estimating the time since bleaching or dyeing. Means we can identify the time since bleaching or time since dyeing with the uncolored portion of the hair. If it is 1 mm uncolored portion of the hair, then it should be around 1 month before the person was bleached his hair. This is also a previous question in UGC net. And a curly appearance with constrictions in the shaft indicate permanent waving. With permanent waving, hair will get curly appearance. Then examination under scanning electron microscope can reveal thickly applied hair sprays. Then methylene blue infiltration test used for bleached and permanent waved hair. This is also a UGC net previous question. Then detection of semi-permanent dyes. This may be detected on hair by TLC, thin layer chromatography, helps to detect semi-permanent dyes. Then detection of oxidative dyes and other hair care products. Usually GC, gas chromatography and GCMS, gas chromatography mass spectrometry have been used to detect hair care products. And the combination of GC, GCMS and TLC can help in discrimination of hair dyes more efficiently. Hair damage. If a hair was a normal hair, then it may not split easily. But if it was a cut hair, then it can split very easily because cut hair was not completely covered with the cuticle. So it can get split easily. Then burnt hair shows a brittle charred appearance at the point of burning, and that burning was depends on the temperature. Extent and type of damage depends on the temperature. And in dead people, a feature is visible that is called post-mortem root banding. It is an opaque band 0.5 mm above the root bulb, can be observed with transmitter light microscope. It was shown only in the dead people, and this is called post-mortem root banding. Then sex identification from the hair. Sex can be determined from root sheath by forensic DNA analysis. Yes, because in DNA it is having STRs, short tandem repeats. If Y STRs are present in the DNA, then it belongs to the male hair, and if there is no Y STRs, then it belongs to the female. In labs without access to forensic DNA analysis, means without doing DNA analysis also, they can identify the sex of the person from the hair. There is two staining techniques: quinacrine dihydrochloride staining and acetoarysin staining. Quinacrine dihydrochloride staining is for Y chromosomes and acetoarysin staining for X chromosomes. With these two staining techniques, they can identify the sex of the person from the hair. Then age. Age identification, difficult to determine age from hair, but experienced hair examiner can occasionally determine that as the hair belongs to very old person or it belongs to very young person based on some features like medullary index, size of pigment granules, darkness of color, amount of cortical fusi, and size of unpigmented area just above the root. With these features, experienced hair examiner sometimes identify whether the hair belongs to very old person or very young person. Hair diseases. There are some hair diseases. I added few diseases here. Monolithrix. The hair shows beaded appearance. I also adding few diagrams for understanding purpose. And polytartid. Twisted hair. Then Trichorexis nodosa. Hair shows frayed nodes. Trichorexis invaginata. It shows like bamboo hair. Then trichonodosis. Knotted hair. And pili-annulate, ringed hair. These are the few hair diseases, and next going to the few other terminology of hair like alopecia. This is the common baldness caused because the normal follicular activity ceases. Then telogen effluvium. Because of some psychological factors or diseases, hair is directly transforming from anagen stage to telogen stage, so that it is called telogen effluvium. Means actively growing hair is directly transforming to the telogen stage, so it is called telogen effluvium, and it is because of some psychological factors, diseases. We can see it in the cancer chemotherapy. Side effects of other diseases like cancer chemotherapy are the instances where hair is shed from the follicles which are still in the anagen phase. Here in this video I explained many things about hair, forensic hair evidence, identification, examination, differences between human and animal hair, and identification of different features of human hair like age, sex, race, diseases. I hope this video definitely helps you.